All right, let's start the next presentation. The second to last overgraduate finalist is Freiburg, and I'm very interested to hear about their unicast system. Thank you.
The next generation gene control, what does that mean to you? Well, we have been thinking about this for quite some time. While today's biological problems are getting more and more complex, it is also becoming increasingly difficult to often find adequate solutions towards complexity itself. How can we get a more holistic access to the genetic state of cell differentiation? Latest RNA technologies only gave us a first glance into whole cell transcriptomics. But will we, on the other hand, be able to build up our own synthetic transcriptome one day? Maybe. Within the past month, our team has been working ambitiously towards a first step into this long way that lies ahead of all of us. We were looking for a complex solution that should be most easily handleable. And now, we want to share a tool with you. A tool that allows us to gain activation or repression at nearly every single gene locus of personal desire. We want to share the first item kit with you that allows us multiple gene regulation at once with the smallest amount of imaginable effort. And on top of it, you will be able to conduct this with your own light remote control. Just like watching Cell Biology Discovery Channel in front of your own microscope, whenever you want, with high definition in human cells. This and much more is Team Freiburg's 2013 Universal Toolkit. This is the Unicast Toolkit for gene regulation. And with these words, I'd like to kindly welcome you to our presentation.
The idea of artificial gene regulation is to fuse an effector domain that can regulate gene expression to a DNA-binding protein. This DNA-binding protein can be engineered to recognize a certain target sequence. But then the problem is that you have to alter the protein structure for every target sequence you want. So we wanted to develop a versatile tool that allows us to regulate multiple genes at once and without having to change the protein structure. So we searched in literature for such a tool, and we found the CRISPR-Cas system. This system consists of mainly three parts. So we have a DNA-binding protein, the Cas9, and two non-coding RNAs, the tracer RNA and the cre-RNA. So this cre-RNA contains a 30-nucleotide spacer, and this spacer actually is complementary to the sequence of your target gene. So simply by base pairing, the Cas9 protein can be directed to its target gene. The tracer RNA thereby mediates the interaction of cre-RNA and Cas9 protein. So Cas9 originally comes from a bacterial immune system, so its natural function is actually to cleave DNA that it recognizes. So the first thing we had to do to use this as a regulatory protein was to mutate the nuclease domain of Cas9. So thereby we created a catalytically dead Cas9 version. And to this protein, we could then fuse different effector domains to regulate gene expression. So now, by introducing any customized spacer sequence, we can direct this whole complex to our target sites without destroying the DNA and regulating it with whatever effector we fuse to the Cas9. So we wanted to standardize this great system for the IGEM community. So as a template for the standardization, we used the Cas9, a human codon optimized Cas9 version. And this included also two nuclear localization sequences. This is very important because we want to direct this bacterial Cas9 protein into the nucleus of mammalian cells. Also, it included an HA tag, which allows us to later detect our proteins in Western blots and other assays. So to standardize the huge Cas9 protein, which has about 150 kilodaltons, we had to mutate out in total 10 restriction sites that were illegal for item standard. So this was quite an effort, but we achieved it in the end. But of course, we also want to standardize the RNA components of the CRISPR-Cas system. So therefore, we constructed a second plasmid, which contains the loci for the tracer RNA and the Cre RNA. So if these two plasmids are expressed, we obtain the catalytically dead Cas9 protein and the tracer and the Cre RNA that can form the complex to bind DNA. So the second plasmid, actually, we call the RNAmer plasmid. This name is derived from the goal of this plasmid to guide the Cas9 to our aims in the DNA. So you can see here a schematic of the RNAmer plasmid, and actually the target site for your customized gene you want to target can be inserted just by using annealed oligos, which is very cheap, by ligation. So you can imagine you can enter different target sites into different RNAmer plasmids, and because we also constructed everything in the iGEM standard, we can use the iGEM assembly method to combine different target sites in only one plasmid. So therefore, we only have to transfect one plasmid with one Cas plasmid, and therefore can direct the Cas9 protein to different target sites at the same time. So this allows us to really multiply target genes simultaneously. So now we've shown you how we constructed basically the heart of our project, a catalytically dead Cas9, which can be fused to an effector, and also the RNAmer plasmid, which allows us to multiply target loci. So we transfected these two plasmids into mammalian cells and assayed their function. But now we will show you what the effectors actually were about and what results we achieved with this in the past month.
So, as mentioned before, we wanted to alter gene expression in mammalian cells. And the first thing we wanted to do is to activate gene expression. To perform this, we fused one of the best characterized and well-known parts of synthetic biology to our Cas9 protein, which is the VP16 activation domain from herpes simplex virus. So, when coming in close proximity to a promoter, this domain is able to stimulate the gene's expression. Well, let's have a look at the results. We performed a reporter gene assay where we transfected a reporter gene called ZEAP, which is a small secreted alkaline phosphatase, which can easily be measured in the supernatant of mammalian cells, as well as our Cas9 VP16 and a plasmid which was coding for the corresponding RNAs. On the y-axis, you see our readout of the reporter gene. And the first thing we did was to transfect plasmids for the protein, the reporter plasmid, but no RNA at all, to see what is the basal level of our reporter. Well, then we started targeting our Cas9 VP16 to different loci in front of the promoter by transfecting RNAs that were complementary to the sites. And we achieved an increase of 3-fold or even 10-fold in comparison to basal level. But one of the main advantages of the Cas9 system is the ability to target several loci at once. And, well, we simply transfected those two RNAs in form of plasmid, which yielded us an enormous increase of expression, which is more than 25-fold. As soon as we had the first data for our activation system, we modeled the system to see where are the bottlenecks, which is the limiting factor in our system. And we realized it was the expression of the Cas9 VP16. This led us to the choice of really strong mammalian viral promoters, like the CMV promoter. Well, as we were able to activate gene expression, the next step was to repress genes. And to do so, we fused a histone methyltransferase to our Cas9 protein, which is called the G9A. G9A has the ability to transfer methyl groups to histones when coming in close proximity, thereby altering the chromatin structure and densing the chromatin and shutting down expression, basically. We've been looking for a test locus where we can assay this, and we found the VEGF locus. VEGF stands for vascular endothelial growth factor, a secreted protein which can easily be measured in the supernatant of the cells. Well, as we were planning this experiment, we had a major concern that the repressive effects we would observe would not be due to our catalytically activity of the histone methyltransferase, but to the sterical block a big protein binding to the DNA might perform. Well, to rule this out, we not only can transfected Cas9 G9A, but also Cas9 without any effector, to simply see if this makes a difference. We transfected into cells these constructs as plasmids, the RNAs, and we targeted them to the endogenous VEGF locus. On the y-axis, you see the readout of ELISA-based method, and on the x-axis, you see the different sites that were targeted. And at first, we didn't target it anywhere, so just letting it float around in the cell. Well, and we didn't see any difference in expression of VEGF. But as soon as we started targeting it in front of the transcriptional start site of the endogenous VEGF locus, we were able to achieve 50% repression in VEGF expression. And this is really cool, because VEGF, the vascular endothelial growth factor, is one of the key players in tissue engineering, which make the system really interesting for application. But here we have a repression using an epigenetic mechanism. But there are other repressing mechanisms as well. And so we fused another repressive domain to our system, which is called the CRUB domain. This protein, when coming in close proximity to DNA, may recruit repressive factors for efficiently repressing transcription. And as we were able to repress the endogenous VEGF locus with G9A, we wanted to try this as well with CRUB. And as you can see, again, we were able to obtain a 50% reduction of VEGF. But we wanted to see the repression with our own bare eyes. And to do so, we performed a fluorescence assay. Here we simply transfected a constitutive GFP, and as you see, we have quite a strong fluorescence. But as soon as we co-transfecting Cas9 CRUB, as well as plasmids coding for the RNA, targeting it into the promoter of the constitutive GFP, we barely see any fluorescence at all. So we were really efficiently shutting down expression of this GFP. To sum it up a little bit, what we have now is a set of working effectors. But do you want to have your genes activated or repressed all the time? No. There will always be occasions where you want to have your gene only repressed during a certain developmental stage, for example. And we've been thinking, what could we do to make our system inducible somehow? And the most elegant way to do this, we thought, would be using light. Well, we've been thinking how could we achieve a light inducibility of our system? And we found a system in plants. We've been taking a step back and split our system again into its two main components. On the one hand, the DNA binding protein, Cas9, and the effector domain, VP16 in this example. These two main components were fused to proteins coming from Arabidopsis thaliana's light signaling pathways, Cry2 and CYP1. These two protein domains have the ability to interact when illuminated with blue light. So the Cas9 binds to its DNA. As soon as the system is illuminated, VP16 coming in close proximity to the Cas9 and therefore may be able to stimulate gene expression. To test this, we used again a reporter gene assay. So we've been transfecting plasmids coding for the Cas9 Cry2 and CYP1 VP16 construct, as well as corresponding RNAs. And again, a ZEAP reporter gene. And again, on the y-axis, you see the reporter readout, and the white bars indicate the plates that were kept in dark, whereas the blue bars represent the plates that were illuminated with blue light. As long as we didn't target our Cas9 to any locus, to any DNA, we didn't see any significant induction at all. But as soon as we started targeting it onto 4 sites in front of the promoter simultaneously, we were able to get a 5-fold induction of gene expression. Well, this may not be as efficient as the Cas9 VP16 itself, but it is a proof of principle that our system may be induced with light stimulus. And we did not only construct systems for blue light, we did construct systems for red light as well, and for UV light. These systems are working as well, but there are still further challenges that have to be faced concerning efficiency here. Well, I've been showing you a lot of molecular biologist data by now, but we wanted to characterize our system a little bit more in vitro, a little bit more biochemical. And to do so, we developed an assay, which we call the unicast binding assay, or UniBase. This is basically an ELISA-based method to assess the binding capacity of our Cas9 proteins, and even the fusion proteins. Well, all you need to have is a 96-well ELISA plate, which is coated with streptavidin. Now you can simply add oligos that are biotinylated and have the corresponding target sequence in it. Now all you have to do is to add the Cas9 protein or the expected fusion protein and the corresponding RNAs. These are then able to bind, and now it's simply an ELISA using a first antibody and a secondary antibody which is coupled to an enzyme and can give us a color change. And this color change can easily be measured colorimetrically and give us an impression how the binding affinity of this Cas9 protein is. Well, as mentioned in the very beginning, we had to mutate our Cas9 protein from a DNA cleaving protein to a DNA binding protein. And we wanted to know if these proteins can still bind as good as the original one. Well, we've used this assay to do so. If I may show you some data. So, on the y-axis, you see the amount of bound Cas9 protein. And the first thing we did simply to code an oligo which did not have the corresponding target sequence, so our Cas9 should not be able to bind to it. And we don't get a high readout here. But as soon as we were coding the correct oligo, we get a high readout for as well the original Cas9 protein, for as well as the catalytically dead variant which we've created. And as you can see, there's no difference in the binding affinity of those two proteins. Well, our protein may bind as good as the original one, but Cas9 has a main disadvantage. This is rather a really big protein. So we've made several approaches to cut this down, to reduce its size. And, well, this is really last-minute data, but we have a promising candidate that may be working and is 630 amino acids smaller than the original Cas9 protein. But the data is still inconclusive here, but we will investigate here further till the Boston Jamboree. Well, so far for the results.
So, does everybody know what time it is? Yeah, it's tool time! Okay, just to give you a short summary, I'd like to present you the Team Freiburg's Unicast toolkit. But what does it include? Of course, the heart of our toolkit is standardized Cas9. But also the Unicast activator for tuning your genes. But on the other hand, also the Unicast repressor, as well as the Unicast histone modifier for downregulation. Our special joker, the Unicast RNAmer for multiple targeting at once. A remote control to induce your systems by different light stimuli. And last but not least, the UniBase to assess the DNA binding capacity of your Cas9 fusion constructs. But why don't you test everything on your own? I mean, it's really, really easy. With the help of our online manual, which is our new way of sharing, you will be able to design your own Unicast experiment, step by step. Everything you have to do is to choose between the effectors. Then decide if you wish light induction or not. After that, enter your target sequence into our Cree RNA design tool, and you will receive several oligo recommendations that could fit with your experiment. In the end, you will receive a customized instruction that will guide you to our experiences, your goals, and success. Everything you have to do is to use our online manual kit, order the biobricks at the registry, synthesize the oligos for a couple of euros, and follow the instructions. You could even clone new effector domains to our standardized Cas9 and thereby expand our Unicast toolkit. But this is up to you.
What do we hold in our hands? It's actually quite simplistic, right? Just these two tiny RNAs, just one single protein, Cas9, guiding us to most of our purposes and desires. But there's so much more behind it. What, if not this standardized Cas9, would fit in so elegantly with the concept and ideal of synthetic biology and iGEM, with modularized, flexible, and easy domain exchange? Indeed, regarding our experiment, we were using components from bacteria, an activation domain from a virus, a histomodifier from a mouse, an open reading frame terminator from a cow, and an incredible light switching system from the rich kingdom of plants. And all of these components were shown to efficiently interact together in one system, in human cells. And precisely because of Unicare's simplicity and enormous power, we have also sought the outer laboratory spheres. We wanted to reach both young and old, went to visit schools and radio stations, and we also went to an international philosophical conference discussing our ideas and visions, and also the risk of such a project. But even the leaders of the most critical political German party in terms of genetic engineering were keen to listen to our ideas, and were very likely discussing with us. What we realized, and what is most important concerning such a project, was that responsibility and communication always strictly belong together, just like our RNAs and Cas9. So now you, what would you choose from? Would you do Boolean logics or a censoring technique? Would you like to do tissue engineering as mentioned before? You can do that. Add domains by yourself if you want, and use it for your own purposes. So actually there's just one last question left for us all. The next generation gene control, what does it mean to you? And with these words, I'd like to kindly thank to all of our sponsors, especially the Biological Center for Signaling Studies in Freiburg and Promega, to our supervisors and advisors who are sitting here, and in the name of this whole fantastic team, finally I'd like to thank you all for your attention.